biology, the chapter Biomolecules. Our today's topic of discussion is enzymes. You know what are enzymes. You have studied in your lower classes, okay? And you know what is a catalyst in a reaction. What is a catalyst? In your ordinary inorganic chemical reaction, we are using catalyst. Is it not? What is a catalyst? You have learned this definition in by heart. A structure which is used in a chemical reaction in order to increase or decrease the speed of the reaction without undergoing any change. Okay. Anyway, a catalyst in a reaction has a role to increase the speed of the reaction. Okay. Whereas in our body, number of reactions take place. When I talk or when I even sleep, a number of thousands of metabolisms or metabolic reactions takes place in a living organism's body. Okay, almost all of these reactions are catalyzed reactions. In those reactions, catalysts take major role, take part in that reaction. Without the catalyst action, the reaction rate will be very slow. So, understand, in living organism's body, almost all metabolic reactions are catalyzed reactions, and the catalysts in biological reactions are called enzymes. So enzymes are named as biocatalysts or biological catalysts. So what are enzymes? Enzymes are the catalysts used in biological reactions. So they are known as biocatalysts. Then what are enzymes chemically? You have studied in biomolecules, you have studied protein, lipids, okay, carbohydrates. What are enzymes chemically? Chemically, enzymes are proteins. Most of the enzymes are proteins. Whereas some RNA can also act as enzymes. When RNA acts as enzymes, that are known as ribozyme. Okay, so enzymes and ribozymes. Is it clear? So enzymes are biocatalysts. Enzymes are present in our body to catalyze almost all reactions in our body. Different specific enzymes are there. Okay, even if when our glycolysis, Krebs cycle, multi-step metabolic pathways are there. A gives B, B gives C, C gives D. In all these steps, there are different enzymes required. Okay, in a metabolic pathway even, all steps are catalyzed by enzymes. Okay, so enzymes are biocatalysts. Most of them are proteins and some RNA can also act as enzyme. They are called ribozymes. Okay, then what happens in a reaction? In a chemical reaction, what is normally happening in a chemical reaction? We take some substrates and either the substrate combine to form a product or the substrate dissociate to form two different products. Okay, either one substrate split to form two products, otherwise two substrates join to form a product. Anyway, in a chemical reaction or in any biological reaction, substrate gives product. Okay, S gives P. Then how we calculate the speed of the reaction? If it's a chemical reaction or a biological reaction, what is the speed of the reaction? The speed or rate of the reaction is the amount of product formed per time. Okay, delta P by delta T. How many products are formed in a particular time? That is the rate of the reaction. Okay, then what is the role of enzyme here? The role of enzyme in the reaction is it gives the more number of products per time. Okay, that means by the action of the enzymes, there are more products formed in a particular time. So, enzymes enhance the rate of the reaction. Okay, so what's the role of enzyme in a reaction? It enhances the rate of the reaction. It increases the speed of the reaction. Means more products are formed per unit time. Okay, so that is what about the role of enzyme. You can just understand the role of the enzyme in our body with this particular example, biological reaction. It takes place in our RBC blood. Carbon dioxide plus water gives carbonic acid, H2CO3. In order to transport carbon dioxide as bicarbonate ion, this reaction takes place in our RBC. Carbon dioxide plus water gives carbonic acid. And this reaction is catalyzed by the enzyme Ca. In short, it is known as Ca, carbonic anhydrase enzyme. Okay, suppose we are just demonstrating this reaction outside, not in the living body. We are demonstrating in the lab. Okay, in the absence of the enzyme, if this enzyme is not present, if it's not given, about 200 molecules of H2CO3 are formed in an hour. Within 1 hour, just 200 molecules of H2CO3 only formed. Okay, whereas if we provide this enzyme, it produces about 6 lakh molecules of H2CO3 in 1 second. You just imagine the role of enzyme there. The enzyme is increasing the speed of the reaction about 10 million times. So this is what is the role of enzyme in our body. Okay, with enzyme, the rate of the reaction is very high. Okay, so that is what is a good example is given here with carbonic anhydrase enzyme. Without carbonic anhydrase enzyme, what happens for that reaction? Okay, the same enzyme is required for the reversible reaction also. I mean bicarbonate split to carbon dioxide and water. You will study in your other chapter. Anyway, this is what is to understand the speed of how the enzyme influences the speed of the reaction. Then what happens? How can the enzyme how is this enzyme able to increase the speed of the reaction? What magic it does? How is it possible for an enzyme to increase the speed of the reaction? What is actually the enzyme we have studied is mostly protein, and the protein in the tertiary structure, it has many folds, and in this folds it has some pockets, and in these pockets it can accommodate the substrate. So these pockets are called active sites. In the tertiary structure of the protein or the enzymes, there are so many crevices which can be called active sites. These active sites are specific for fitting with the substrate. The substrate can be fit to the then it enhance or facilitate the bond formation or bond breaking, whatever is the reaction, whether it's breakdown, catabolism or anabolism, it speed up that process. Okay, so by accommodating the substrate in the active site, an enzyme can speed up the catabolic action or what? Anabolic action. Okay, then the role of enzyme action can be given in a graphical representation. It gives you a better idea how an enzyme increases the speed of the reaction. Okay, just listen to this graph. In a reaction, in a biological reaction, substrate gives product. Okay, normally when the substrate gives product, before reaching into the product, the substrate has to pass through a transition state. Substrate is giving product, in between there is a transition state. Okay, the energy required for the substrate to reach the transition state is called activation energy. The energy difference, substrate level, transition state level. Okay, the energy required to reach the transition state is activation energy. Okay, suppose the first here I have given a red colored graph and representation with the black color. In the first case, the substrate and products are shown here. In the first case, the black graph, substrate is giving the product and here is the transition state. In order to reach the transition state, an energy is required. That without that enzyme, here suppose in the first graph there is no usage of enzyme, there is no involvement of enzyme. So the substrate requires more energy to reach the transition state. It takes time to reach the transition state. So that the product form they get delayed. Okay? I mean the number is less. Whereas in the second graph, the red colored graph shown here, this is an enzymatic reaction. So the enzyme lowers the energy, activation energy. So this substrate can reach the transition state very quick and it can give the product very quick. This is what is an enzyme doing in a reaction. Okay, in a biological reaction, the substrate has to pass through transition state before forming the product. Okay, the energy required to reach the transition state is called activation energy. With the help of an enzyme, the activation energy is lowered. It requires less time to reach. Okay, so more easily products are formed. Whereas without enzyme, the activation energy is not, it requires more energy, so that product formation is delayed. Less number of products only formed. This is what the interpretation of the graph. So what the enzyme do? The enzymes lowers the activation energy required for a substrate to reach the transition state, so that it quickly reaches the transition state and quickly gives the product. Okay, so the enzyme is giving the product. The substrate is giving the product easily with the help of the enzyme. Okay, this is activation energy without enzyme. With energy it is lower. So the red color graph is with enzyme. Then the black graph is without enzyme. Okay. The same idea is conveyed in a graphical and cartoon representation here. What happens at the time of enzyme action? At the time of enzyme action, enzyme at its active site, which is specific for the substrate. Substrate only can come and bind there. It is highly specific, structurally specific for the accommodation of substrate. The enzyme at its active site binds to the substrate. Enzyme plus substrate gives an enzyme substrate complex. This is short-lived. It immediately gives a very short-lived enzyme product complex. Then that is split to enzyme and products. It is given in a cartoon representation here. Enzyme, its active sites. Okay, specifically the substrate with special kind of structural modification, correct fitting to the enzyme. So forms enzyme substrate complex, short-lived, immediately forms the two products. The bond is broken. Suppose the bond is broken, two products are formed. It is initially attached to the enzyme. So EP, enzyme product complex, very short-lived, immediately dissociate and make the enzyme free, and the two products are easily formed. Okay, this is what is the cartoon representation of enzyme action. Enzyme plus substrate gives short-lived enzyme substrate complex. This enzyme substrate complex immediately forms by bond breaking or bond forming. It forms an enzyme product complex, immediately dissociates to enzyme and products. Okay, that's about the cartoon representation. You have one more small topic. What are the factors affecting enzyme action? The major factors affecting enzyme action. You know, enzymes are catalysts. If when iron, other metals used as catalyst in inorganic reaction, you know, when we increase the temperature, it will be, the speed will be double or like, okay. Whereas here it is not like that. If enzymes are most of the reaction, in most of the biological reactions, enzymes are the catalyst. Okay, enzymes are proteins. It cannot withstand high temperature. It gets denatured at high temperature, and also at high pH it cannot act. So enzyme actions are favored at optimum temperature and optimum pH. Okay, beyond that the temperature is increased or pH is increased, it decreases the enzyme action. That's what is shown here. Suppose this is the optimum, this is temperature, enzyme action. This is the optimum temperature. When temperature increases up to this, okay, the rate increases. But after optimum temperature, mostly in our body case, 37 degrees Celsius temperature. After that temperature, it gets lowered. Enzyme action gets lowered. This is the graph. Similarly for pH, at optimum pH, up to optimum pH, and the pH increase can favor the enzyme action. Whereas after that, it is lowering enzyme action. Okay. Whereas in the case of, it's not giving this type of action for substrate concentration. Suppose in a biological reaction, we are adding the carbon dioxide and water. We are adding more and more carbon dioxide and water, means it increases the rate of the reaction. Okay, until the full enzymes are used. I mean, up to this maximum rate, it increases. Then after it will be steady, because the number of enzymes may not be sufficient for binding to this. We are adding more and more carbon dioxide and water. Substrate concentration is more, but we are not adding sufficient carbonic anhydrase. Then it affects. I mean up to the 100% saturation of the enzyme. When all the enzymes are occupied or used, then no more increase. After Vmax, maximum velocity, no more increase, no more decrease. So it's a same level. Okay, so this gives a graph like this, only the substrate concentration. The pH and temperature gives a graph like this. After optimum pH, decline. After optimum temperature, decline. Well, after maximum substrate concentration, steady level. Okay, no decrease, no increase. Because there are no more enzymes to get accumulated. Okay, so that's all about the factors affecting enzyme action. Enzymes actions are not usually favored by high temperature and high pH, whereas there are organisms which live in high temperature, hypersaline lagoons and all, okay, high salted areas. Such organisms, their enzymes are thermostable. Okay, they may require higher optimum temperature. If very high temperature also, their enzymes can function because their optimum temperature is high. Our optimum temperature will be our body temperature, about 37 degrees Celsius. Almost all biological actions will be favored high at this temperature. Okay, these are the factors affecting what? Enzyme action. Okay, any more doubt means we will clarify in the live session and we will have a elaborate interaction at that time. And thus today we will wind up. Thank you.